excited to have guests today. We have Amanda Moores and Rebecca Pugh with NIST, National Institute of Standards and Technology, and they are with the Environmental Specimen Bank, and they will share a lot more about that and what they do. So this is our Scientist Spotlight series, and I'm super excited to have you guys on today. Obviously, we have a history. I worked at the Environmental Specimen Bank with these ladies a number of years ago. And they are based in Charleston. So this is really exciting for us to bring a little bit more awareness about the Environmental Specimen Bank to some of our followers here locally. So do you guys mind introducing yourselves and telling us your role?
Sure. Thanks, Lauren, for having us. First off, this is exciting. I'm Rebecca Pugh, and I'm the group leader for the Biospecimen Science Group of the Chemical Sciences Division at NIST. And I have actually, I'm going to date myself a little bit here, have been with NIST for about 22 years now. Which is, yeah, crazy.
Hi, thank you, Lauren, so much for highlighting us and letting us be your scientist spotlight. My name is Amanda Moores, and I am the program coordinator in the Biospecimen Sciences Group at NIST, and I oversee day-to-day operations inside the NIST biorepository. I've been with NIST for, surprisingly, even dating myself, just a little over 15 years, and it's been a great and wonderful experience.
That's awesome. So we're going to just jump right in. You guys said a lot of really big words in your introductions. So could one of you take it and tell us a little bit more about the Environmental Specimen Bank and what it is and what you guys do?
Sure. Yeah, I'll start it off, and anything I miss, Amanda, jump right in. So NIST has actually been archiving samples for a specimen bank for long-term archival. Basically, it means samples are collected in a very standardized way that can be used many, many years down the road for other types of research, retrospective research. And so NIST actually started banking samples in the late 70s, looking at actually human liver samples that were collected with the Environmental Protection Agency at contaminant levels across the US. And when I say contaminants, those are things like organic contaminants, so chemicals in the water, chemicals in your body, organic pollutants, persistent organic pollutants, as well as inorganic trace metals and heavy metals, mercury, arsenic, all those types of things. And so that's really where NIST's involvement started, is and why we're in the Chemical Sciences Division, is because we have a lot of chemists that are doing the analysis of these samples, looking at those contaminants. But what NIST and other collaborators have realized, really the sample that's collected is extremely important and needs to be high quality for that work to be done down the road. Otherwise, if you, as they say, garbage in, garbage out, if you don't have a high quality sample, then your research down the road isn't going to be as high quality either. And so that's how NIST kind of got started. But then we've taken it really to another level of, the science of that biospecimen, of that sample, and how to collect it properly, how to maintain it, and how to archive it for that long-term research.
So Amanda, can you tell us more about the more current specimen bank, like samples?
Sure. So another important aspect of the environmental biological specimen banking, biobanking world is we're not the only ones doing something like this, doing work that involves biobanking. There is a community of biobanks that are part of a professional society called ISBR, the International Society for Biological and Environmental Repositories. And in this society, the groups and specimen banks from around the world collaborate together, share ideas, ask each other questions, talk about what we're doing, showcase our research. Every year we have an annual meeting where we can talk and invite speakers to come and say why we're doing the research we're doing. It's amazing how you can collaborate with a researcher doing human analysis on samples and apply that to the environmental or the animal world as well. what NIST does really well, and where we're brought in, is because of our standards and our protocols. Rebecca mentioned that samples were starting to be collected with the pilot project of the human liver back in the 70s. A lot of those protocols were then put in place to collect different environmental samples, and the samples that we've collected back in the 70s, the 80s, the 90s, those have all been consistently collected in the same manner, so you get the same, ideally the same information. You're not going to introduce, we do our best not to introduce any contaminants, so the sample that is collected is a high quality sample. And for example, we use titanium and Teflon, and we try to be consistent with that with our processing of samples. So it's all very consistent with standardized protocols for our collections.
So how do you keep a sample for 50 years stable and, not adding in, and not losing it?
So great.
Yeah, go ahead.
Great question. Samples can be stored in many different ways. So we all are very familiar with our refrigerators in our homes, and maybe your deep freezer that you might have. We go beyond freezing just at -40. You can freeze biological samples at -40, -80, or liquid nitrogen temperatures. So in the NIST biorepository, we have approximately, it goes in it, 28, 29 liquid nitrogen vapor phase freezers. What that means is we store our samples in the vapor portion of liquid nitrogen. Our freezers are large capacity. They have about ranges between 6 and 8 inches of liquid nitrogen, and the samples are stored in either Teflon jars, plastic cryo vials, Teflon bags, inside standardized and very well organized racking systems, tubes or jars. They're all very labeled. We know where everything is inside our liquid nitrogen vapor phase freezers or -80°C mechanical freezers.
Wow. How cold is a liquid nitrogen freezer?
The max coldest temperature is -196°C. So very cold. We wear special gloves whenever we need to, cryogenic gloves, to remove our samples from the freezer. We also, in our biorepository, we take safety very seriously. The room conditions are monitored for temperature, humidity, oxygen. We want to make sure we maintain the highest standard of safety for everyone working in the biorepository, above all else, making sure everyone working in there is safe.
So you have the samples, so say these liver samples from the 70s. Why is it, you touched on it, but are people using those samples, and what would people be using them for 50 years down the road? Any sample?
Well, and so I mentioned the human livers, but one thing I didn't mention, and it's important because we're here with the Low Country Marine Mammal Network. One of our other projects that we have is the National Marine Mammal Tissue Bank, which is our longest running and current project that we've had in working with NOAA and a lot of other collaborators that collect samples from stranded animals. So that actually started in the late 80s, 1987, up in Alaska. But looking at those samples, and similar to the human livers, is as you archive these samples down the road, other types of research, you might think of other types of research that could be conducted on the sample. But also, as technology gets better and better every year, your instrumentation and your detection limits get more precise. And so sometimes, 20 years ago, you might not have been able to detect a certain contaminant or a certain trace element in that sample because of the instrumentation, or the method wasn't really well developed. Fast forward 20 years, things are much better, and you're realizing that was in that liver sample, or that was in that biological tissue, because you can reanalyze the subsamples, or what we call aliquots, of those tissues that were banked when they were initially collected. In addition, there could be other contaminants that we weren't aware of, or other things in the waters that we weren't aware of, for, like, let's say, the marine mammals, that these samples can then be pulled out of the freezers and analyzed for that, to see if in 20 years ago that particular contaminant existed that they weren't aware of, or there was some sort of contamination of something else that people weren't aware of at the time, but we can go back and look in time, if you will, retrospectively, to look at that. And when I say we, I don't do any of that type of analysis. That's really for the outside researcher or for the NIST scientists that can get access to these samples to run those types of analysis. And in addition, you can do like temporal trends, which are time trends. So you can look at the different levels of these contaminants or whatever you're analyzing for over time, because a lot of our samples are taken from similar, species, or not necessarily the same animal, but the same species every year for the last, 20 years. So you can actually look at those levels over time, or even a geographic region.
Rebecca mentioned a great point, and a great thing about the National Marine Mammal Tissue Bank is when we work with partners from Dolphins here in Charleston for Low Country Marine Mammal Network, we collect liver, kidney, and blubber samples from marine mammals. And we take that one liver and we will split it into two samples. So we've got an A sample and a B sample that are systematically stored in two separate freezers. Additionally, that A sample is going to be stored long-term, hopefully for forever. This B sample is available for researchers to request. Additionally, this 150 grams, most researchers don't need 150 grams. They can say, I need a smaller portion. What we will then do is take that 150 grams of liver, cryogenically homogenize it, which just means basically pulverize it into a fresh frozen powder at liquid nitrogen temperatures to keep it very, very cold. And then we aliquot it into smaller portions, which perhaps takes that 150 gram sample into 35 different opportunities for researchers to be able to request that sample. So we've taken this one liver, split it into two, put this one off to the side, this one B sample into multiple opportunities for researchers to be able to use, so it can do what Rebecca said. Some researcher perhaps in 1992 was looking at one contaminant, and then now in 2021, now they can, there's likely multiple aliquots left from that one liver. They can say, well, my instrumentation is better. I've got a better method of analysis that I can go look for more precise measurements. They can pull another aliquot of that. There's a tissue access policy. There's a review that goes through for research when researchers submit a request for analysis. So the samples aren't just doled out to just anyone. There is a policy to look at, and the research that is done is reviewed, and it's always worthwhile to go through the process to request samples to make these measurements more precise, to get better data, to try out new methods on samples that we have inside the NIST biorepository.
It's so neat just to think that, there's these samples that's basically looking back in time.
But there's also the idea that you can have something available to look forward to. And what I mean by that is if you knew what something was like now, what about industry that's going to develop later? What about an oil spill that's going to happen whenever, in the future? When we have the chance to collect samples now, we're preparing for being able to do analysis in the future too, by putting samples and archiving for future work.
So you can compare to what quote unquote normal is, and compare it to if there's a major event in the future.
So that brings up a good point with the oil spill. It's we're fortunate that we were able to we had samples. If there was an oil spill, we would have samples potentially prior to the oil spill, and then working with partners, maybe collections during the oil spill, and then down the road after the oil spill to see if these animals have recovered from that oil spill. And that's working with a lot of partners that we have. And again, and I mentioned NOAA, the National Oceanic and Atmospheric Administration. We have partnered and collaborated with them for the Marine Mammal Tissue Bank since 1987. And they, have the Marine Mammal Health and Stranding Response Program that you're a big part of with the Marine Mammal Network. And we've been able to work with NOAA and all of the stakeholders and letter holders that respond to strandings, not just locally in Charleston, but across the US, and get samples collected from many different species that, sometimes are very rare species even. And that particular stranding network may have some samples that they've collected from that animal, but we also have that very particular standardized protocol that everyone follows, that we know how those samples were touched, how they were handled, and then once they became frozen in our freezers, we have a tracking system. We monitor the freezers so we know it's maintained at those really cold temperatures throughout its life. We haven't had a freeze thaw of the sample, and we have a lot of the metadata that's associated with the animal, as well as how that sample was processed, which a lot of times, an individual researcher may have that, but once they leave their institution or move on, they don't necessarily carry that information with them, or it doesn't go with the sample. So that's the really nice thing about a specimen bank or a biorepository, is you have all of that collectively over that 20, 30, 40, 50 years. That can be difficult to do if you don't have a real program in place like we do.
Touched on this. It's so important because when things happen, for an example, an oil spill, it's usually, let's start collecting samples now, but we don't have anything to compare it to. And that's always the question is, well, what levels were they at before the oil spill? How do we, and it's so great that you guys are being thinking proactively, because that is so important for future work and, cleanup and just understanding the effects. You have to have those beforehand samples that a lot of people don't have.
Another good thing about when NIST was called upon to participate and do specimen banking for actually for any of our projects, the reason why NIST is brought in is because of our standards and because of the high quality work and because of the security of the biorepository. The samples are monitored with a monitoring system inside of a building with security access that is maintained, and there's a chain of custody that is maintained with all of those samples. So an important part about our collections is that, and I mentioned this earlier, is samples that were collected in 1987 are going to be collected the same way in 2021, 2022, in the future. They're not going to be collected differently. And if there is a change, we document that. We put that in our notes. We put that on our necropsy data or on our processing. We make copious notes about how samples are processed and keep track of our protocols. We don't change the protocols unless it's requested, unless there's some reason why everything stays the same. So those beluga whale samples collected in Alaska in 1987, they're going to be collected the same way with the same protocol next year.
And people don't realize how important that is when you are, looking at anything, trends or retrospectively. I mean, if things aren't collected in the same way, it's just like you're kind of comparing oranges and apples. So that is super important in the science field to have very, very consistent sampling, samples, processing.
And noting those deviations. So we actually had one study, a researcher requested samples over time from, and I can't remember, it might've been beluga samples, and they were going to do some proteomics, new methods in developing proteomics. And they, we had a 10-year series, let's say, of these samples. And one of the years he came back and he's like, this data is not correct. Something's not right with this data. Let's look back at the sample again. And we, when we went back and looked at the notes, we realized that was the year that the samples actually were lost, and they ended up thawing and sitting out thawed for a number of hours or days. I don't remember the whole scenario, but we had that noted in there. And that goes to show you how very precious and, one deviation can cause for this particular type of analysis. It may not be for other types of analysis, but for that particular, with the proteomic study, having that sample thaw and then be refrozen made a difference in that downstream analysis. And so we had that information in our database, and we're like, oh yeah, that was the one that thawed. So that helps them to then, as they're analyzing their data, to determine what's going on.
Can you tell us what other types of samples you have at the specimen bank?
So yeah, so besides liver, kidney, and blubber samples collected from stranded marine mammals, We have the opportunity to work with different partners in the marine mammal field to collect samples from live animals, where live animals are safely captured, samples taken and released back into the wild. And samples coming from those animals range from serum, plasma, whole blood, feces, urine, skin, blubber, milk, and analysis are run. So all different types of analysis are run on all different types of samples. The skin can be used to look at trace metals. It can be used to look at genetics. Contaminants can be looked at in a snapshot from the blubber. The urine, you can there's a researcher at the College of Charleston looking at phthalates in urine. So where NIST has, and what we've developed over many years and implemented, is a standardized protocol on how to collect samples for archiving. So again, samples that were collected at our first health assessment in 1992 are still collected the same way now, and they're archived in the bank. So a good example of why those archive samples were incredibly important, I think 2 or 3 years ago, we for many, many years, we've been working with the Chicago Zoological Society's Sarasota Dolphin Research Program down in Sarasota, and there was a red tide event. And at the time when we collected a blubber biopsy, we always archive a piece of skin from the blubber biopsy. Well, the red tide happened after the health assessment, or it was going on. I hope I don't remember the details, but they needed a piece of skin to do more analysis on what was going on with the red tide. And you could look at, they requested a portion of the skin that we could aliquot off because it was already still frozen, and it had been frozen. We were able to take a piece off from this piece of skin that we had, and there was enough there that we could then send that on for analysis. So we had that piece of sample available to be able to do, again, that future research. We didn't know, we didn't, you don't know, we call it the purpose. What's the purpose? And the purpose of that skin was for archive. We're not sure what we're going to need it for, but let's archive it because perhaps we're going to need it for something else. And this is a great example of using an archived sample for something that we didn't know was going to happen.
In addition to marine mammals, though, we also, our current projects, we also archive seabird egg contents from nests that are located in Alaska and the Pacific Islands region, as well as sea turtle tissues and fluids from the Pacific Islands region, and other marine mammal projects with actually human-owned, if you will, samples.
So speaking of marine, we're going to dive a little bit deeper into marine mammals. We obviously work with the South Carolina Marine Mammals Stranding Network, collecting dolphins here locally, stranded animals, and then often bring you guys in to collect samples, but not only from stranded animals, from also health assessments. Rebecca, do you want to dive deeper into kind of some of the work that's done here in Charleston?
Yeah, well, I mean, we work with, again, with NOAA as really our primary partner, as well as the plethora of other collaborators like the Low Country Marine Mammal Network, to get samples collected during these strandings that happen locally in the area, as well as these health assessments where the animals, as Amanda mentioned, animals are brought on board a boat safely, and a complete doctor's visit, if you will, samples are collected. And so we've been able to be a part of those collections and bank samples from those events, both locally in Charleston, but as well as I've mentioned, all over the coastal U.S., working with other collaborators. But fortunately, being here in Charleston, we're able to be at least a little bit more part of with some of the field work that's actually, we're able to go out with you and the Stranding Network, other coordinators, to assist with those collections of the samples and do the processing following our own protocols personally, so we can actually be out in the field helping out with that part of it as well, which is really a wonderful opportunity to be able to go out in the field. And that's probably one of the favorite parts of my job, that I've been able to be out and helping with the field work, and not just in the lab receiving samples and in the bank receiving samples and archiving them, but really being a part of training people to follow our standardized protocols, and then also processing the samples ourselves.
And for those that aren't familiar, the process is very tedious. Collecting samples, there's special knives, Teflon bags, you know, handling is to a minimum. Either they come out, Amanda, Rebecca, or staff comes out to take it themselves. Then the samples go back and everything is processed in a clean room, you know, with gear on. And so it's a tedious process, but as they've mentioned, you know, it's so necessary to reduce any contaminants that we could put onto the samples, and, you know, and doing it in such a way, your standards are very, you know, top notch, definitely amazing. And it's a process to take it from an animal down, you know, to the freezer. And it is very specific.
So yeah,
You remember it well.
Yeah, I was going to say, Lauren, you remember it very, very well, and you've had you're in a unique position where being the experience that you've had working in the necropsies with NOAA, and then working with us to see, okay, now this is where it goes, and go through all of the NIST protocols with the clean room and the Tyvek suits, and the idea of we don't want to contaminate the sample, and we don't want the sample to contaminate us. Yeah. We want to protect ourselves too, and we also don't want to, we want to minimize contamination of the sample. So that's why we do wear, and we process our samples in a clean room environment. We wear the Tyvek suits, we wear the hoods, we pull our hair back, we wear the, we call them special gloves. We just wear the same gloves. We minimally handle the sample. We use very purified water to wash our sample off. And it's the same process that was done, again, back in the 80s. So it's all the same, but with the, you know, with the consistency of, we don't want to contaminate the sample, and we don't want sample to contaminate us.
Mm-hmm. Yeah. And we're really fortunate, though, with the support through NOAA and through NIST, and the support we have, we're able to actually provide kits for all of the samples that are collected. We actually provide the supplies and put together these kits when we train people outside of NIST, obviously, and these collaborators to collect these samples for us. You know, we train them, but we also provide all of the supplies. So that way we know they're not just using this random glove that we are not aware of that can contaminate the sample, or using a different brand of container that they're going to use, or different type of water, which can, you know, cause contamination if it's not the specific. So we're really fortunate that we have that opportunity and that support, that we can provide all of that, because that's really what helps make this extremely successful in the long run.
That's super important to getting people to take, you know, specific samples. It's helpful to have all the gear right there. Yeah. So Amanda, can you tell us, I know you've done a little bit of work, some field work in Alaska with beluga whales, is that right? Can you tell us about that?
Yes. So absolutely, it was a incredible highlight and fascinating work. So I like stories, and I like to tell stories, and so I'll try to tell it as a story, as a you know, how I experienced it. So NIST has been involved with beluga research and looking at beluga whales in Alaska, and we were contacted about the first year that was done was in 2008, and we sent kits up for samples to be collected, and there were 2 opportunities in the spring and the fall where samples were collected. The partnership, the research was a collaboration between NOAA, the Alaska Sea Life Center, Georgia Aquarium, and several other aquaria around the country that had beluga whales. And they had the forethought to say, maybe we should archive some samples. Maybe we should put some samples beside. somewhere for right now. And there was real-time analysis being done on samples too. So in 2008, samples were collected, and then a few years later, they asked if NIST would if we could have someone come up to do the processing. Partly because our processing is very, like we just said, it's very detailed. It's very strict. We have the steps are very tedious. So I had the opportunity to go up to Alaska, and it was an amazing trip. So flew into Anchorage, and then took the logistics involved with doing a health assessment like this there's a lot, because it's a remote location. So then we flew from Anchorage to Dillingham, Alaska, and we're looking at the Bristol Bay population of beluga whales. That population of beluga whales is an increasing population. It's not declining as compared to the Cook Inlet beluga whale population. The whales that live in the Cook Inlet, there's not really a clear reason why their numbers are not increasing. So the beluga whale, the Bristol Bay population was the group we were going to study and take samples from. So I remember going, and I vividly remember when we were in the boats and what the other partners that were there, the Alaska Department of Fish and Wildlife, people from Alaska. So there are native people that will come out on the boats with us that are hired to find the whales and catch them for us, because we don't know how to do that. You're using local expertise and their knowledge, and having them be involved firsthand with the research that's being done on their animals. So they would we had each 2 people on our boats. There were only 3 or 4 of us on it, maybe 4 or 5 people on a boat. So it's very bones crew going out to catch these massive animals. So I remember being in the boat, and we made, the water is it's murky, it's not clear, but we turned this corner, and I remember looking up and like, wow, the water's really choppy. We're going up there. Like, there's a lot of whitecaps, but okay. And as our boats got closer and the hum of the engine, those whitecaps disappeared. And this looks up, I'm like, those weren't whitecaps, those were beluga whales. It was this majestic realization of, it's the first time I'd ever seen a beluga whale. I'd seen pictures. I hadn't even seen them in an aquarium, but to see that, oh my goodness, there are hundreds in this small bay, and they've all just gone for a dive because they heard the engines. And it was wonderful. It was amazing. It was an amazing experience. And then a whale was identified. The men that were with us identified a whale, and were able to safely capture, with using a net, safely capture the beluga whale. And one of the biggest differences between catching the beluga whales and catching perhaps a bottlenose dolphin was the size. They're very, very large animals, and the tides in Alaska are very dramatic. So we had to work a lot with the tides and what time to go out with the boats, because the tides can increase up to, I think in some places, 18 feet. So we had to be very conscious of the tides and very conscious of how long we had hands on the animal. So as the tide went down, the idea that, well, did the animals move around a lot? Didn't they thrash? Didn't they? Well, they did, but they're weightless in the water. So when they're swimming in the water, they don't know how heavy they are. But then when the tide goes down, gravity does what gravity does, and the whales are like, so I think I'm really heavy. I can't move very well. So we use buckets of water to keep their their skin wet, take our samples, do what we need to, wait till the tide comes back in, and then as the tide comes in, and it does very quickly, the animal will then swim away, and we've been able to now collect. And I can't even describe the number, the amount of data and samples, what they're worth to be able to collect from the animals. So since 2008, we've been able to collect over, I think we're at about 60 samples from 60 animals and tag animals, archive samples, and those are archived in the NIST biorepository. So the skills that were used. So I mentioned the Alaska Sea Life Center and the Georgia Aquarium, because what they have done is use techniques perfected in an aquarium setting on human managed care animals and applied those skills to the wild caught animals. So there weren't, techniques that were just, let's try this on this wild animal. They were perfected inside a controlled environment, inside an aquarium, and then applied. And the animal handlers that did that had tens of years of experience doing this. So lots of experienced people with world-class veterinary care from the Alaska Sea Life Center, with native people there that have a vested interest in the animals. And another reason that these samples are so important was Bristol Bay and Dillingham. That area is the sockeye salmon capital of the world. So it's a large fishing community. Additionally, there is proposals to put in a copper mine very close to where these animals live. And with mining comes contamination. And so if this mine were to be put in the Bristol Bay area and contamination would happen in the environment, we have archived samples from the area to see this is what we can do. Analysis can be done in a temporal trend, in this is what it was like before, here's what it's like now manner.
That's amazing. What a cool experience.
Yeah.
I don't know.
Well, I didn't even talk about the human side of it and what we just, the fangirling, geeking out over, I kind of did about seeing these animals. And then, because then we have to go back now, and what we just said with what NIST does is now we go back to the house or the lab and we process those samples, and the work can go on for hours, making sure samples get processed, get stored. And in remote areas, the logistics can make or break a sampling operation. So we had to figure out, how are we going to get liquid nitrogen dry shippers? How are we going to get liquid nitrogen, dry ice, and enough to last us 2.5, 3 weeks? We had to deal with the weather, freezing rain, then hot the next day, and the temperatures of the water. So we wore dry suits that kept us dry in the water, because you can't get in a wetsuit, hypothermia will happen. So there's all those other things in addition to seeing whitecaps. No, those are whales. You're in this dry suit, in this, sitting in a folding lawn chair inside of a boat with kits of coolers surrounding you, and all you want to do is just see these amazing creatures and realize like the wonderful opportunity that is literally in front of you.
Yeah, it's something it's hard to believe unless you've done it or seen it, and the work that goes into it, the partnerships, the amount of people, hours, set up, months of preparation, months of sampling after. It's a lot of work, but a cool experience for sure.
Yeah, I mean, I think just even with any stranding, it takes a team effort and not just one institution or organization to not only get samples, but get information about these animals so that we can learn learn more about them and when their conservation and all these different opportunities. And so, yeah, it really is a huge team effort across all of these different types of collections that I think really is what makes it. And that's the wonderful thing too, is you get to meet so many different people from so many different organizations and institutions that and learn about what their research is specifically, but how that contributes to the whole of the animal and the population and the species and on and on. It's amazing.
Right. It's a team effort with a similar goal, but so many different ideas, research. I mean, I imagine an animal could probably go into, who knows, 30, 40 different research projects, right? So it's,
yeah. And everyone has their own, it's always great to hear about everyone else's passion in what they want to do and why they want to do it. And then how can NIST help? What can we do? What expertise can we provide, and what we can we help with? Rebecca mentioned like another important aspect of it is we're part of a team. It's not just me and Rebecca. We have a team of other NISTers that work with us in our group inside the NIST biorepository that bring their own expertise and their own passions, and like you did, Lauren, and you compare notes and you want to be able to help, and that's what I hope we're able to do. And what we are doing with the stranding groups is what can NIST do and work by working with partners and with the rest of our team to do research, to maintain samples, to help success happen.
Yep. Is there anything else you guys want to share with the public that we just don't know or is important to get across?
I mean, when you working with you and with the NIST biorepository, I would like to say we are, as far as locally here in Charleston, we're located at the Hollings Marine Laboratory on Fort Johnson. Unfortunately, we don't really do like public tours, but if you go to the NIST website and type in NIST biorepository, you can, we've got pictures up there. There might even be an older video where you can see the inside and see all of the freezers and everything to get a better understanding of what the biorepository looks like. But we're always willing, as Amanda mentioned earlier, a lot of our samples, there are tissue access policies, so researchers could use these samples for their own research. But for like the general public, just a thank you to them, to everyone for, helping you, for volunteering and being a part of the Stranding Network as a volunteer, spreading the word, learning more about these beautiful creatures that we have in our own, waterways here, as well as, all over the U.S. And the globe, really. And I'm just feel very fortunate to be a part of NIST and be a part of setting up this biorepository down here in Charleston. And as Amanda said, we have an amazing team of researchers that work both in our group, in our division, at NIST, and then as well as all of the collaborators that really contribute to the success of this biorepository.
One thing I'd add is to, like Rebecca said, we don't do public tours at the lab, but that doesn't mean that the public can't be involved. And I think one of the good thing that we've got is what you're doing, Lauren, is the public education with the Low Country Marine Mammal Network, being involved in dolphin count. And the idea of making sure that, the dolphins that live here, the marine mammals that live here, they're locals. This is their home too. When I've talked to the public and said, this is where they live, it's this idea of, I said, like you live at your house. This is where they live. This is their home. They don't go to North Carolina. This is their home. They stay here. They live here. People that live, on the water, they see those animals. They know them. They live in our local waterways. So we should do what we can to protect them by not littering, not harassing marine mammals, not throwing away your fishing line, staying away from marine mammals, the safe distance, and just, watching these majestic creatures from afar and protecting them and encouraging children to do the same, to have these practices put in place for lifetimes, and really appreciating and respecting the locals that live in our waterways.